Welcome again to our workshop entitled Application of NGS in the Diagnostic Lab on Archival FFPE Tissues. And it's our honor to welcome this afternoon Dr. Brian Walker from the Royal Marsden Hospital. Brian received his Ph.D. from the Imperial College in London. He was focused on the co-evolution of antigen processing genes with MHC molecules. He then moved to the Institute of Cancer Research as a staff scientist, where he focused on using a lot of different molecular techniques on identifying genetic determinants that can be used to classify myeloma. And then after that moved to the Royal Marsden Hospital, which is basically the partner of the ICR, as a senior research scientist, where up to very recently he led a translational research team focused on implementing new technologies, so especially NGS and then also ddPCR to improve molecular diagnostics of cancers. And then probably 2 weeks ago, I guess, Brian moved to the US, moving back to his previous work on myeloma. He's now the director of research at the University of Arkansas Myeloma Institute.
Thank you very much for that introduction. It's a pleasure to be here today, and thank you for staying around to the end of the day to listen to this presentation. So as Marique said, I've kind of moved recently from the myeloma field into diagnostics in FFPE tissues. This was a bit of a shock to me, moving from high-quality DNA from a blood sample into low-quality DNA from an FFPE sample. And so we've done a lot of work over the last year and a half to try and improve the process by which we can use the FFPE DNA and NGS technologies. I've kind of worked in different areas of NGS over the last few years, and the way I see it, there are kind of three different ways of looking at this. The first is a discovery phase using perhaps whole genome sequencing or exome sequencing. This is usually for research purposes and is used for identifying new targets and characterizing diseases. So you have a set of samples in your freezer that you're interested in analyzing. They're not related, not part of any clinical trial, but they might be your rare samples and you want to find out what mutations or abnormalities are present in these samples. The second is more of a translational field. So you might be using exome sequencing or a targeted panel. Again, mostly for research purposes. You might want to use this to investigate the impact of new targets. So you might have a set of related samples from a clinical trial, and you want to sequence these and find out if any of the mutations are related to prognosis. And then the third is more of a diagnostic panel, a focused panel for diagnostic purposes. And then this would be used for clinically relevant abnormalities, reporting data back to the clinic, which may result in altered treatment strategies for patients. So at the Royal Marsden in London, we have 5 panels covering different diseases. These are a GI panel, a pediatric tumour panel, breast tumour panel, hematology panel, and a lung panel. At the moment, we have 2 of these being used in clinical trials, the reports of which go back to the clinic. That's the GI panel and the breast panel. Our GI panel is our original panel. We've been using Kappa reagents for sample prep and then Nimblegen reagents for the capture. The GI panel was our first panel. Design wasn't great, but we've done some work with that to improve it. And then we've moved on to pediatrics and breast and the hem-onc and lung as well, which are in operation too. And then these are all sequenced using the Illumina MiSeq in the diagnostic lab. These panels must be able to detect certain things that we're interested in, such as SNVs and indels, translocations in the diseases where they're relevant, and also copy number abnormalities. The thing that we do, which I don't see a lot of other places doing, is that we also want to determine if a gene does not have a mutation. So we want to know if a gene is wild type, and I'll come back to that later in the presentation. So working with solid tumours, we have several challenges with the sources of material. So this is an H&E slide from a sample, and you can see here that we're often dealing with very small biopsies from which we're expected to extract DNA and perform our tests, which obviously can be problematic. And then looking at these samples in more detail, we can see that even though there are some sample there, it's not all tumour. And if we get closer and closer, you can see that in actual fact, a lot of it is not tumour. We also have problems with sample cellularity. And so we found that we can characterize samples according to the cellularity within the biopsies. And this can help us see in advance whether or not a sample will work in the sequencing. And so we could have a sample with high cellularity. We've got lots of cells here going down through intermediate and low and very low, where even though we have some sample, there may not be particularly many cells there to use and extract DNA from. Then we also have challenges with DNA quality. So the tissues taken are often fixed with formaldehyde, and the fixation method that is used can often affect the DNA quality at the end. We often see that samples that are coming from different hospitals within a network often behave in a different way, and this usually depends on how the samples were fixed in the first place. And obviously, the fixation with formaldehyde can lead to excessive cross-linking and DNA degradation, and also deamination artifacts as well. So looking at DNA integrity, we found, again, that the best way of looking at this is using the Biorad tape station. They've more recently introduced genomic DNA tapes or screen tapes for this piece of equipment, and introduced also the DNA integrity number. And this, you're probably more familiar with the RNA integrity number, or RIN. And this is a similar thing for DNA integrity. So samples which you'd expect perhaps from the blood or hematology samples have a high molecular weight and a high DIN number. And as the DNA is fragmented, more and more the DIN gets lower and lower. We often find that this is a good indication of how well a sample is going to work in the sample preps. So we started off by using the Kappa HyperPrep kit before moving on to the Hyper Plus kit. So I'll just take you through this briefly. So we extract DNA from our samples using QIAGEN kits for the FFPE and also the blood DNAs. We fragment the DNA using the Cavaris with the HyperPrep kit, and then go on to do end repair and A-tailing and adaptive ligation, where we add on the indices for the samples. Perform a pre-high PCR of 8 cycles, and then qubit each of the amplified samples at this point, and do sample pooling. So we normally pool 5 tumour and blood pairs per sample pool. We actually tend to put the tumour DNAs in a separate pool to the blood DNAs, because we find often the blood amplified DNA tends to outcompete the FFPE amplified DNA in the hybridization. We then take these sample pools into the Nimblegen capture, so using whichever panel is appropriate for the tissue type. We started off using 2 overnight hybridizations with the Nimblegen panel. This is mostly because our first GI panel was badly designed by us. We had a high off-target rates, and so to overcome that, Nimblegen suggested we do 2 overnight hybridizations. We since then optimized the panel designs and have gone down to 1 hybridization. And we find that we get a slightly lower on-target rate with 1 hyb, but we also get a lower duplicate levels, and this balances out. So we can save a day's work basically by removing that hybridization step. After hybridization, we do another post-hyb PCR, qPCR the pools, and then put them on the MiSeq for 2 × 75 base pair paired end reads. So the Hyper Plus protocol is pretty similar, just with a few changes shown here in red. So the fragmentation is not done by the Cavaris, it's an enzymatic fragmentation. There's 1 or 2 things to be careful of at this point. So the enzymatic fragmentation is very susceptible to EDTA concentrations, so it's important to know how much EDTA is in your elution buffer from the DNA. There's a conditioning solution that comes with the kit, which is basically calcium chloride, I think, which collates out the EDTA. And if you add too much or too little, then the amplification step doesn't work quite so well. And often blood samples and FFPE samples are using different buffers, and so it's just worth knowing which kit you're using, which buffer your DNA is in. After that enzymatic fragmentation, there's no end repair step, and we go straight onto the A-tailing. And again, adaptive ligation for the indices. And then at the pre-high PCR, we now only do 6 cycles. We find that the A-tailing adaptive ligation is more efficient with the enzymatic fragmentation, and so we actually get higher yields with the Hyper Plus kit. And so we can get away with using fewer PCR cycles at this stage. And then the rest of the process is the same as for the HyperPrep kits. So we've used both the HyperPrep kit and the Hyper Plus kits extensively now. I think we've probably used Hyper Plus kits on somewhere near 500 to 1,000 samples now. But we did perform some validation of how the comparison was between the Hyper and Hyper Plus, or rather Cavaris and Hyper Plus enzymatic reaction. And what we found was, looking at the mean panel coverage at 100x and 250x, we ended up with a higher percentage at 100x and 250x for the Hyper Plus samples for both FFPE and blood. Not a massive difference at 100x, but the Cavaris fragmented samples didn't perform so well, and so they drop off quicker. And so the Hyper+ samples have a better depth, and so we end up with better coverage at 250x for both FFPE and blood. We also find a decreased number of duplicates in the Hyper+ samples compared to the Cavaris. You'll probably notice that these are quite high. These were when we were doing 2 hybs, and so we had higher duplicate levels. Now when we do 1 hyb, it's probably down around about 20% for the duplicates in our panels. We also had increased unique on-target reads with Hyper+ compared to Cavaris for both FFPE and blood samples. And all of these things combined means that actually our median panel depth shot up in the Hyper+ prep samples compared to the Cavaris. This was especially true for the FFPE samples. We didn't see quite so much of an increase for the bloods, but for the FFPEs, the enzymatic fragmentation compared to the Cavaris was a massive improvement in the overall quality of the samples, resulting in higher sequencing depth across the panels. So as I said, we have 2 studies at the moment running in the clinic, whereby we report results from our panels back to the clinic. The first one is format study. This aims to recruit 200 patients with locally advanced metastatic or GI cancers. We are collecting archival samples or repeating a biopsy if required, and then performing sequencing of samples using a panel of potentially actionable mutations, translocations and copy number changes, and aiming to get the results back to the clinic within 3 weeks. So that 3 weeks is from when the molecular diagnostic lab receive the FFPE slides to producing the results. We then discuss these at a sequencing tumour board meeting, whereby patients are either stratified into potentially suitable treatment, depending on what mutations they have, or if there's no molecular target, they continue with their standard treatment. So we have several QC points during this process. So within our lab, from the H&E stain slides, we look to see if there is actually tumour present. When we often look at these H&E slides, we sometimes find that there is no tumour at all within them, and so we reject them outright there and then. Where there is tumour present, we then look to see what the tumour cell content is in the sample, and again, stratifying that according to percentage of tumour cell content. And then looking at the cellularity of the sample, this is high, intermediate, low. And if the samples pass through the H&E slides QC stages, we then extract the DNA, and if there's enough DNA extracted, we sequence the samples. At the moment, we're using 200 nanograms of DNA for these samples. So just some graphs of the influence of different things. So the first one is the influence of specimen type. So we often find that resections work a lot better than biopsies, often because we have more material. And so these fails come from mostly the H&E QC steps where there is insufficient material. And again, looking at the H&E slides, we fail a lot of samples based on their tumour cellularity, mostly when they're low, and then less frequently at intermediate, and those with high generally pass. And also looking at tumour cell content, so those with intermediate or high, so greater than 30% tumour cell content, generally do well and go on to get sequenced, whereas those with low tumour cell content fail QC early on. And then going back to the DNA integrity number, we can see that from our samples, the ones that actually went on to be sequenced, you can see how bad the quality is of most of our samples. The scale being from 0 to 10, 10 being the best. Very few of our samples actually get halfway up the scale. Most of our samples are very poor quality, but we still manage to get good sequencing data out of those. We run matched samples, so we run tumour sample and then also blood from the same patient. Initially, when this trial was set up, it was set up not to have a matched blood sample, but we quickly got that changed. This is mostly for an analysis point of view. It's so much easier to do your analysis when you have a matched blood sample from the same patient. And just to show that, there's been a few papers out recently this year showing analyses with paired and non-paired samples. This particular paper here says that without a matched control, about 30% of candidate somatic mutations are actually germline SNPs. You can do some bioinformatic teasing out of the data using non-somatic, well, potentially SNPs using databases such as dbSNP, or conversely looking at COSMIC and trying to find out which mutations have appeared in COSMIC and could be somatic. But in general, this is ineffective, especially for large tumor suppressor genes, where you may have a large number of private SNPs within patients, making it difficult to decide whether or not a particular mutation is somatic or not. And this generally reduces the specificity and sensitivity of your assays. I'm sure you all know, but just in case you don't, a paired sample analysis looks like this. So you do the sequencing on your blood sample, sequencing your tumour sample, identifying variants from both samples, perform a subtractive analysis, and identifying somatic mutations which are only present in the tumour, in this case, the TP53 mutation. And looking at one of our samples in particular, we performed an unpaired and a paired analysis. And so from the unpaired analysis, we identified somewhere around about 11 or 12 different mutations. This is in a panel with 46 genes, about 200 kb. Some of these could potentially be somatic, and you may have to go and search through databases to try and figure out which ones of these were actually potentially somatic. But when you do a paired analysis, it's very easy. You get 2 mutations out at the end. One's a frame shift, one has a stop gain. These are very clearly somatic mutations. And it makes reporting of the data in the clinic much easier and more robust. So I said earlier that we were also interested in reporting wild type. This is important in colorectal cancer, but it's probably important in other cancers as well. People have found that in colorectal cancer, if you are wild type KRAS, treatment with cetuximab, which is an anti-EGFR antibody, results in a better overall survival compared to just best supportive care alone. But if you have mutated KRAS, cetuximab has no benefit on patient outcome. And so there's no point giving cetuximab to a patient with a mutated KRAS. So how do we determine whether a gene is wild type? So this is a calculation that's based on the depth of the sequencing done, the tumour content, and the percent of the region of interest that is covered. So we're basically aiming to get at least 100 reads from the tumour. But if we only have 50% tumour, then our minimum depth jumps up to 200x. And then if you have a higher tumour percentage, your minimum depth will come down, and if you have a lower tumour percentage, your minimum depth will go up. Most of our samples turn out to be a lower tumour percentage, somewhere around 30 to 40%, and so we're aiming for a higher minimum depth. This is across the entire gene, so each exon within the gene, we want to have that depth. So we look at the percentage coverage for each gene, and if that's greater than 95% at our minimum depth, according to tumour percentage, we say it's high confidence wild type. If it's greater than 85%, we say it's medium confidence. And if it's less than 85%, we fail the gene. And the kind of report that we generate at the end, we don't just report the variants we find, we report everything. So for each of the genes in our panel, we give a result. So we have each of the genes, the method we're using, the regions of interest, maybe all of the exons. It may be specific exons within a gene which are more relevant. And then we have the gene result, the variant allele frequency, and then the gene report, which says whether a variant is present or whether it was wild type or failed, and whether or not the test is complete. That's pretty hard to see, so in a little bit more detail. So for example, if KRAS, if we identify a variant, we have the C dot and P dot nomenclature as shown, and a report would say a KRAS variant. If no variant was detected and greater than 95% of all of the exons within KRAS that are captured have a sufficient depth according to the tumour content, then we say it's a high confidence with no variant detected. If it's greater than 85%, we say a medium confidence. And if it's less than 85%, we would fail the results for KRAS and not report at all. So because we work in a diagnostic lab, we have to validate each of our panels and the process as a whole. And so we have to determine our limits of detection, sensitivity, specificity, and reproducibility. So the GI format panel was the first one we did. And so we compared the data with a set of samples which had been analysed by other technologies in the past, such as Sanger sequencing, SSCA, or COBAS. So we sequenced 58 samples in total on the format panel, covering about 150 mutations, which were derived from these various platforms. We use a 5% variant allele frequency limit detection, and from this we were able to get a 96% sensitivity and a 98% specificity with 95% confidence intervals. This was quite expensive and time-consuming to do, and so with our other panels, having now validated our process as a whole, the only thing that's changed now is each panel that comes with the different tissue types. And so to validate the panels, we're now using cell blends from Horizon Diagnostics. We use 4 different cell blends, which have known variants within them and known variant allele frequencies. We run each of these 4-cell blends twice with different operators, different sequencing machines, so that we can assess a whole variety of different variables. The first being repeatability. So looking at how repeatable the samples are, we have pairwise correlation of SNPs of about 0.98 and a correlation of indels of 0.67. Indels are always harder to call, and so you always get a bit lower. We have looked at the breast panel again using sensitivity and specificity, and we find for SNPs we have a sensitivity and specificity of greater than 99 and 98% with 95% confidence intervals. And for indels, sensitivity and specificity a little bit lower, at greater than 95% and greater than 81%. I should say that we actually do detect all of the known SNPs and indels in the samples. It's just that sometimes there aren't that many of them that fall into the intersection between our panel and the ones described by Horizon. And so we end up with lower sensitivities and specificities at the 95% confidence interval level. We can also look at accuracy. So they have a series of validated SNPs or indels with known variant allele frequencies, which they've determined by ddPCR. So we can correlate the sequencing variant allele frequencies with the ddPCR and find we have a correlation coefficient of about 0.98, which is very good. One of the more important things, especially again going back to when we want to call wild types, is panel uniformity. What we mean by this is that the panel captures in a uniform way across all of the regions that we're interested in. We don't want to have some regions being overcaptured and other regions underperforming, not capturing, and always failing. So here's an example of a good panel. This is our breast cancer panel. What we've done here is look at all the regions and the normalized depth of each of the regions of interest. And that's been normalized against the mean depth across the entire panel. And you can see we have a nice narrow peak around 1, showing that most of the regions capture uniformly. If we look at each of the genes, we can see that most, again, of the genes coming out at the same depth across the panel, with the exception of 1, which seems to be amplified in these cell lines. And then we can compare this with a bad panel. So this is our first GI panel, which was less robust. And so you can see here that it's not as uniform as the breast panel. We have lots of regions which underperform down at this end. And you can see these here, different genes which don't capture quite so well. And so these always fail, which is a pain. We're holding back the clinic. They're always asking us, why do these fail all the time? And it's because the panel wasn't very good. We've since optimized that. And the second version of our GI panel now looks more like this. So that's mostly CNVs and indels. So going on to detecting translocations, this is quite important in some of the leukemias and also sarcomas, whereby certain translocations can identify which diseases you're dealing with in the clinic. So how does this work with capture technology? So you have your fragmented DNA, either enzymatically or by Covaris. This obviously contains fragments from all across the genome, as shown by the different colors. And some of these fragments will be hybrids between different chromosomes where translocation takes place. So all you need to do is perform your normal sample prep methods, and in your hybridization capture, you will have baits designed against your region of interest. Being a myeloma guy, I like the IGH locus, and so we often capture the IGH locus to capture translocations in myeloma. So if you're doing that, you would have sequences against the IGH locus, which would then enrich for these gray fragments which contain your region of interest. And again, some of these gray fragments also have parts of other chromosomes attached to them where the translocation is. We then sequence the fragments using paired end reads. So each of the fragments gets sequenced at both ends and align the sequence to the genome to identify fragments with sequences from different chromosomes. So this fragment, both ends will map to chromosome 14. But in this example, we have one end mapping to chromosome 14 and the other mapping to chromosome 4. And so we can determine this sample has a T414 translocation. So I've used this in myeloma quite a lot in the past, and bringing this into the solid kind of domain, we've used this in several cancer types. So in lung cancer, we've looked at the EML4 ALK rearrangement. So this is actually a large inversion happening within chromosome 2, but it's the same principle. All we're doing here is capturing the region of interest in ALK. So I think this is right between exons 17 and 20, perhaps. It's the end of ALK anyway. So we just tile everything across there pull down the entire region, and then bioinformatically look to see if there are any translocations or inversions, which it's very difficult to see by the eye, but the programs can pick them out, and it's actually these kind of teal coloured reads here. And when we go to the pairs of these reads, we can see that within EML4, we have a bunch of reads which are showing up here, showing that we have an EML4 ALK tumour. And this is important in lung cancer because these can be treated with crizotinib. We can also do this with Ewing sarcoma. So the handy thing about this is that we have one gene and many partners. So EWSR1 partners with many different genes in Ewing sarcoma, but we can detect all of these just by capturing EWSR1 introns 6 to 9, and that will pull down anything that's attached within those introns. So a couple of examples. So we have EWSR1 CREB1 translocation. So here we have the region of interest on EWSR1, and you can see that in some of these regions, we're not getting any pull down at all. This is usually because of repetitive sequences. And so although we may be capturing these, the bioinformatics won't realign them to the genome confidently. So you do stand a chance of missing some translocations in this way. But here we found some reads which have partners which map to CREB1, showing we have an EWS CREB1 in this sarcoma. And again, the same region in EWSR1 being captured, but in this sample, we identify reads going to FLI1. So that's a different translocation, but using the same area. So the last thing that we are interested in is looking at copy number changes. So having worked in genetics for a while, I always kind of look back to the good old days of SNP arrays. I still think they're probably the best way of determining copy number in samples. You can get very nice, clean data from your samples, as is shown here. But you can do it using NGS. The more data you have, the better. So from exome sequencing, we can easily detect, this is a myeloma sample, so we can detect the common deletions in FAF1 or FAM46C, as well as gains in 1Q, or deletions of 17P, or interstitial deletions of TP53. But with FFPE samples, especially on panels, the data is much noisier. So here, this is a very simple plot where we've normalized the number of reads that we're getting in each region of interest against the mean panel depth for that sample, and also against the normal sample for that patient, so that we can try and use the number of reads that are captured as an indication of the copy number. And you can see it's very difficult to try and tell anything from this. Here we do have a low-level amplification of ERBB2. It's about 5 copies there, so it's about 2.5 on the scale. We have to double that up to 5 copies in a 40% tumour, so it's probably about 12 copies in the actual tumour. And we did actually confirm this by ISH. But you can easily detect amplifications in FFPE samples. So here we have an example of a sample with both EGFR1 amplification and cyclin D1 amplification. So we have all of the exons within EGFR1 were amplified, and cyclin D1 as well, equating to probably 60 copies of EGFR1 and about 22 copies of cyclin D1. Often when we see these samples with high levels of amplification of any gene, it normally negatively impacts on the rest of our panel, because these amplifications are basically stealing reads from all the other genes. And so when we can identify these amplifications, usually the rest of the genes fall below our thresholds of detection and end up failing. This isn't a problem of these small panels. So for fresh frozen samples, the copy number data is much nicer. So here are 2 pediatric tumour samples, which we've looked at as well. And here you can see a much clearer distinction between copy number breakpoints. Here we've got a CDK6 gain and ATM deletion, or another CDK6 gain and P10 deletion. So it really is a problem with the FFPE samples not performing quite so well in this way. And so to try and get around this, we have stolen the ideas from the SNP arrays that we've used in the past. So what we've done in designing our assays or our panels is not to just include the exons, but also to include SNPs within the genes which have minor allele frequency greater than 0.4, so that we can use these SNPs as an indication through allelic imbalance whether we have gains and deletions. And obviously the more SNPs you have, the better your data ends up looking. So in the breast panel, we've done this, and if you just use read depth, you can see in this sample it's actually not bad. So here you can see some amplification of MYC. We have deletion of CDKN2A and B, ATM deletion, a normal BRCA2 and RB1 on chromosome 13. And then if we look at the SNP allele calls within that same sample, so in the top here, we have the germline data from the blood, B allele frequencies. So here we have on the outsides of this plot are the homozygous calls. So we have homozygous A, homozygous B, and in the middle are the heterozygous AB calls for the blood, and then the tumour. And if we look at these, so on chromosome 8, you can see here in the blood, these lots of heterozygous SNPs, which disappear in the tumour, and we have gain in the number of reads at that locus as well, showing that we have the MYC amplification at that particular point. Again, on chromosome 9, we have heterozygous in the germline, losing these heterozygous reads going out towards homozygous, and also loss of copy number showing the deletion of CDKN2A and B. And then on chromosome 13, we have the heterozygous calls for BRCA and RB1, which are lost in the tumour, but we retain normal copy numbers. So here we're actually identifying copy number neutral LOH of BRCA and RB1 on chromosome 13. So this can be a nice way to, or a second way to help kind of justify the copy number that you think you're calling. Then I'd just like to finish there and thank the people within the Marsden who have done that, my team within the Marsden, and then David Gonzalez and his team within molecular diagnostics, as well as the units that have been involved with our samples and the trials as well. Thank you.